Put two people from opposite sides of the world beside each other, and the visible contrasts may seem enormous, yet their genomes are overwhelmingly alike. The traits our eyes notice most, skin color, hair form, and parts of the face, are controlled by a limited set of biological pathways. They are highly visible, but they are not a measure of how biologically separate two people are. Homo sapiens is a young species. Fossils attributed to early members of our species are roughly 300,000 years old, and most ancestry outside Africa descends substantially from populations that left Africa much later. That is not enough time for separate human subspecies to form, especially because populations repeatedly met, traded, migrated, and had children together. Genetic difference is also distributed in overlapping gradients. A gene variant may become gradually more common from one region to another, but there is rarely a clean line where one biological population ends and another begins. Mountains, deserts, and oceans can slow movement, yet none created permanent walls around all human ancestry. So this episode will not ask why separate races evolved. Biology does not support that picture. It will ask a more precise question. How did ultraviolet radiation, temperature, food, thin air, chance, and interbreeding alter particular traits in particular populations? Each mechanism left a different kind of mark, and none changed the basic unity of our species. Human skin color depends mainly on the amount, type, and distribution of melanin produced by melanocytes. Humans with different skin tones have broadly similar numbers of melanocytes. The crucial difference is how actively those cells manufacture pigment, how large the pigment packages are, and how long those packages remain inside surrounding skin cells. Melanin is made inside tiny structures called melanosomes. These packages move from melanocytes into keratinocytes, the main cells of the outer skin. There they form protective caps around cell nuclei. The pigment absorbs and scatters ultraviolet radiation before part of that energy can damage DNA or disrupt sensitive molecules. When early humans lost much of their body hair and became active in equatorial Africa, exposed skin faced intense ultraviolet radiation. Dark constitutive pigmentation, pigment present even without tanning, provided persistent protection. One proposed selective pressure was protection of folate, a B vitamin important in DNA, asynthesis, sperm production, and early embryonic development. Ultraviolet radiation can break down folate compounds, although the exact contribution of folate protection to pigmentation evolution remains debated. The key point is that selection acts through reproduction. A trait spreads not because it is aesthetically useful, but because, on average over many generations, carriers leave slightly more surviving children. Farther from the equator, ultraviolet B becomes weaker and more seasonal. That matters because UVB starts vitamin D production in skin. A cholesterol-derived molecule is converted into vitamin D3, which is then processed by the liver and kidneys into an active hormone that helps regulate calcium and phosphate. If too little vitamin D is produced during childhood, bones may mineralize poorly. Severe deficiency can cause rickets, soft, deformed bones that make movement difficult and can alter the pelvis. In environments with weak UVB and limited dietary vitamin D, variants producing less melanin could allow more UVB to penetrate and increase vitamin D synthesis. This did not produce one universal switch from dark to light skin. Different populations evolved lighter pigmentation through partly different gene variants. Migration and intermarriage later mixed those variants. Skin color is therefore a repeated evolutionary response to local UV conditions, modified by diet, clothing, and ancestry, not a permanent label for a continent. Temperature can influence average body proportions because heat is exchanged across surface area. A compact body has less surface relative to its volume, so it loses heat more slowly. A long, narrow body exposes more surface relative to its mass and can release heat more efficiently. This is a physical tendency, not a rule for every person. Anthropologists often describe these patterns with Bergmann's and Allen's rules. In many warm-blooded animals, colder climates favor greater body mass and shorter extremities, while hotter climates favor longer limbs. Human populations show parts of this pattern, but nutrition, disease, workload, and childhood growth can strongly alter an individual's final body form. Hair shape is produced inside the follicle. Straighter hair tends to emerge from more circular follicles, while tightly curled hair grows from more curved follicles, and the hair shaft bends as it forms. The genetics are complex. Many variants affect keratin structure, follicle shape, and growth, and similar visible hair forms can arise through different genetic routes. Tightly curled scalp hair may help reduce solar heat gain while still allowing air circulation and sweat evaporation. Experimental models support this possibility in hot, sunny conditions, but hair evolution was probably shaped by several pressures, including insulation, moisture, and sexual selection. A deep version must preserve that uncertainty rather than turn a plausible mechanism into a proven single cause. All human infants normally produce lactase in the small intestine. Lactase sits on the brush border, the microscopic surface of intestinal cells, and splits lactose, the sugar in milk, into glucose and galactose. Those smaller sugars can then cross the intestinal wall into the blood. In most mammals, including most humans historically, lactase production declines after weaning. If an adult then drinks fresh milk, much of the lactose remains unbroken and passes into the large intestine. Bacteria ferment it, producing gas and drawing water into the bowel. The result can be bloating, cramps, and diarrhea. Pastoralists did not simply wait for evolution. They changed milk first. Fermentation converts part of the lactose into lactic acid, and cheese-making removes much of the lactose with the whey. Archaeology finds dairy residues and milk proteins thousands of years before lactase persistence variants became common. Culture created a usable food before genes caught up. Lactase persistence occurs when regulatory DNA near the LCT gene keeps the lactase switch active after childhood. Different pastoral populations evolved different regulatory variants. That is convergent evolution. Similar biological outcomes produced by separate mutations, because people in different regions created similar milk-dependent ways of life. Why did the variants spread so strongly? Milk supplied water, calories, fat, and protein. But simply drinking milk may not explain the whole selection signal. During famine or intestinal infection, lactose malabsorption could worsen dehydration and diarrhea. Adults able to digest fresh milk may have gained their largest advantage precisely during crises, when a small survival difference mattered most. Milk therefore changed biology through a feedback loop. Herding created a new food. Fermentation made it partly digestible. Regulatory mutations allowed some adults to digest fresh milk. In communities where milk became especially valuable, those adults left more descendants. The cultural practice changed the environment in which natural selection operated. Starch is built from long chains of glucose. Digestion can begin before food reaches the stomach because salivary alpha amylase cuts those chains into smaller carbohydrates. The enzyme is encoded mainly by AMY1 genes. Humans vary unusually in how many copies of AMY1 they carry, and copy number often influences how much amylase appears in saliva. More gene copies do not guarantee perfect starch digestion because pancreatic enzymes, gut microbes, food processing, and individual regulation also matter. But populations with long histories of starch-rich diets often show higher average AMY1 copy numbers than populations relying less on starch. Recent ancient genome research suggests strong changes in the amylase region continued after farming expanded. Cooking was part of the mechanism. Heat gelatinizes starch, water enters the granules, their structure loosens, and enzymes gain easier access. Grinding grain breaks protective plant tissues into smaller particles. Humans did not adapt only by changing genes. They repeatedly changed food mechanically and chemically before digestion even began. At high altitude, the percentage of oxygen in air remains about the same, but atmospheric pressure falls. Each breath therefore delivers fewer oxygen molecules to the lungs. The immediate response is faster breathing and a faster heartbeat. Over days, the kidneys release more erythropoietin, or EPO, stimulating the bone marrow to make additional red blood cells. More red blood cells can carry more oxygen, but too many make blood thicker. Thick blood raises resistance inside vessels and increases strain on the heart. Long-term high-altitude adaptation is therefore not simply the ability to produce enormous amounts of hemoglobin. It is the ability to balance oxygen delivery against the dangers of excessive blood viscosity. Tibetan populations carry strongly selected variants near EPAS1 and EGLN1, genes in the hypoxia-inducible factor pathway. This pathway acts like an oxygen alarm. When oxygen falls, HIF proteins activate genes involved in breathing, blood vessel responses, and red cell production. Tibetan-associated variants blunt parts of this response, helping prevent hemoglobin from rising excessively. Some EPAS1 ancestry in Tibetans is closely related to DNA from Denisovans, an archaic human population known largely from genetic evidence and a few fossils. Interbreeding supplied useful variation. Natural selection then increased it. Adaptation did not always require waiting for a brand new mutation. Humans could acquire an existing variant from another human lineage. High-altitude evolution happened differently elsewhere. Many Andean highlanders show higher hemoglobin concentrations than Tibetans, while Ethiopian highlanders display still other combinations of oxygen physiology. The environment may be similar, but the available starting genes, population history, and time at altitude differ. Evolution often reaches several workable solutions rather than one perfect design. Not every population difference is an adaptation. Imagine that 20 people establish an isolated settlement. By chance, they carry only a sample of the genetic variation from the larger population. A rare variant may be absent, or it may be unusually common among the founders. Their descendants inherit that accidental starting pattern. This is a founder effect, a form of genetic drift. Drift changes variant frequencies through random survival and reproduction, especially in small populations. It does not improve the body. A harmless trait, a disease-causing variant, or a neutral blood group allele can all become common simply because the population began small and remained isolated. Bottlenecks work similarly. If famine, disease, or migration sharply reduces a population, the survivors do not carry every variant at its previous frequency. When numbers recover, the population may remain genetically different from its ancestors. Selection is about consistent reproductive advantage. Drift is statistical chance amplified across generations. Isolation was rarely permanent. Ancient DNA repeatedly shows population replacement and mixture. Steppe pastoralists moved into Europe, farmers expanded from Anatolia, Bantu-speaking communities spread across large parts of Africa, and Austronesian sailors crossed the Pacific. Each movement carried genes, languages, technologies, and diseases into new combinations. Interbreeding with Neanderthals also left functional effects. Most people with substantial ancestry outside Africa carry roughly 1 to 2% Neanderthal-derived DNA, although not the same fragments in every person. Some inherited variants influenced immunity, skin biology, or metabolism. Others were removed over time because they reduced fertility or functioned poorly in a modern human genome. The result is a web, not a ladder. A visible trait may reflect adaptation to sunlight, another may be a random founder effect, and a third may have arrived through migration. Even traits in the same person can have completely different histories. Appearance is therefore an unreliable shortcut for total ancestry, ability, character, or biological worth. Why do humans look different? Because populations experienced different ultraviolet levels, temperatures, foods, and oxygen pressures, because small groups carried accidental samples of variation, and because populations repeatedly mixed.